thanks goes to the chair for introducing me and the academy for giving me the opportunity to present my work here. So I am presenting some of my work in 20 minutes time and trying to give an overview of what we do in our lab. So the title says that our lab works with mouse hepatitis virus, which belongs to the beta coronaviridis family and genus beta coronavirus like SARS-CoV-2. And this virus, when we use it as an experimental animal model, it causes a complex disease mechanism which is demyelination and axonal loss. And this demyelination and axonal loss mimics certain pathological hallmarks of human neurological disease, multiple sclerosis. So just to give you an overview of multiple sclerosis is a chronic, progressive or relapsing and remitting demyelinating disorder that affects the central nervous system and can lead to severe physical or cognitive disability as well as neurological deficits. This disease is much more prevalent in Northern Europe, Northern America, Australia, New Zealand, but in India it is an orphan disease. It ranks as a major cause of nervous system disability in young adults aged 20 to 40. And in multiple sclerosis, the myelin sheath, actually in our nerve fiber, we have a concentric ring surrounding the nerve fiber which is called myelin sheath. That is a protective membrane and that helps the nerve impulse to go from one nerve to another nerve very fast, and that's what it causes saltatory conduction, and if there is a inflammation then that disease happens. So, to tell you that central nervous system by itself is a very complex organ. It has different cells like neuron, astrocytes, oligodendrocytes, microglia, ependymal cells, and all different cell types are arranged in a complex interacting system. They have very specific role to play and they interact with each other through metabolic coupling and then perform the function of central nervous system and the peripheral nervous system. So, as multiple sclerosis, the name says multiple, there is a multiple etiology, multiple cause, multiple disease mechanism and multiple pathology are involved. So, to tell you, like, first, if there are some of the neurons are myelinated and there is a, due to the myelin sheath, actually this nerve impulse go from one nerve to another very first. That's what it is called saltatory conduction. It jumps from one node to another node. But if it is non-myelinated nerve fiber, nerve impulse goes slowly. But in different diseases, body generates autoreactive T cells to myelin proteins, and these myelin reactive T cells cross blood-brain barrier and attacks the myelin sheath and causes autoimmune demyelination. While autoimmune demyelination is very much known, there are neuroinflammation progressively started to denote chronic CNS cell specific like neuron, astrocytes, microglia and oligodendrocytes inflammation that can also cause the disease. So you can see here, this is the myelinated nerve fiber and the intact myelinated nerve fiber looks like that. This is the axoplasm and surrounding the concentric rings, but in some diseases there are lack of this concentric myelin sheath, that is called hypomyelination, or if there is a complete removal, that is called demyelination. So demyelination is a pathological hallmark of human neurological disease, multiple sclerosis. That's what we are, multiple sclerosis to study in human, it is very difficult, but we are trying to understand in an experimental model system. And this multiple sclerosis causes meningoencephalomyelitis, optic nerve inflammation, there are vocal changes, there are bladder function difficulties, and there are several other symptoms happens. Why? Because people believe body generates autoreactive T cells to myelin protein. Myelin reactive T cells cross blood-brain barrier and neurological deficits attribute mainly to demyelination, and there is a permanent neurological disability in MS have been attributed to neuronal damage and loss of neurons, and brain atrophy also seen in MRI, and histopathological changes, as I said, meningoencephalomyelitis and optic nerve inflammation, optic neuritis including axonal damage are one of the prominent disease process. And the estimated number of people that with MS worldwide has increased to 2.8 million in 2020, but in 2013 it was like much less. 2020 global prevalence is 35.9 per 100,000 people, And if we try to understand the etiology, there are several school of thoughts. People believe it is mainly an autoimmune disease because body generates it T cell, as I mentioned here, autoreactive T cells, which goes to the brain, crossing the blood-brain barrier and attacks myelin sheath. There are like school which believes that there are viral infection and there are genetic factors also involved. So all these things makes it a multiple sclerosis. So to understand the autoimmunity, we have an experimental animal model which is called experimental autoimmune encephalomyelitis. For understanding the viral infection, we have Thailand murine encephalitis virus, TMAV, or mouse hepatitis virus, which will be the focus of my talk today. And there are genetic and environmental factors, and for that we have several knockout mice model and transgenic model. So we try to understand the complex disease mechanism in an experimental animal model. To understand that, first thing, what happens, as I mentioned, body generates autoreactive T cells, it's attack myelin. If myelin sheath is destroyed, axon doesn't know how to wrap around. That's what there is a damage from myelin sheath, which is outside, to axonal loss, which is inside. So it is an outside in model, and one of the example is experimental autoimmune encephalomyelitis. Whereas in intact myelin, if the virus propagates through the axon, axon degenerates, and if axon is not there, axoplasm is lost or there is a damage, then myelin sheath doesn't know how to wrap around. So that time it is called inside out model. So in TMEV and mouse hepatitis virus infection, axonal degeneration shown to be inside out model, which I will be talking today throughout my talk. So to understand the complex disease mechanism, we have in vivo in intact mouse where we inject with the EGFP engineered mouse hepatitis virus, and we study the histopathology to understand what is going on at the brain and the spinal cord level. As well as we have in vitro neuronal cell culture and the primary glial cell culture to understand in a reductionist model what is going on in individual neural cell types. So to tell you what happens from the beginning, when we inject with the virus, the intracranial inoculation of the virus infects CNS cells, and then they actually activates astrocytes and microglia, which are the CNS resident immune cells, and there are chemokines and cytokines get secreted, and leukocytes starts to migrate to the brain, and if there is an inflammation in the meninges, it is called meningitis. If there is an inflammation in the brain parenchyma, this is perivascular cuffing, and microglial nodule formation, this is called encephalitis. If the virus propagates to the spinal cord, then the inflammation in the myelin sheath called myelitis. At that point, during acute stage, viral RNA reaches its peaks by day 5 to 6 post-infection. Infectious virus particle also goes up by day 5 and 6 post-infection. After that, the immune system, the inflammatory peripheral immune cells started to come, and they started to check the viral replication. So in coordination with the microglia macrophages, the peripheral T cells start to interact and bring the anti-inflammatory response. The viral titer goes down below the detection limit. Viral RNA also goes down but persists to some extent, and demyelination starts as early as day 5 to 6 post-infection but reaches its peak by day 30 post-infection by 4 weeks. And you can see here, this is looks all fast blue stain spinal cord. There is a lack of myelin sheath, that's what it is called demyelination, and there are concurrent axonal loss also. So MHV infection in mice induces biphasic acute neuroinflammation and chronic demyelination and axonal loss. So to do that, the part I am trying to say today, that we very recent studies, in several studies, we tried to dissect the role of important immune checkpoint proteins in regulating viral induced neuroinflammatory demyelination. So among the new checkpoints, what we did, we looked at the EFID2. When we looked at the EFID2 both at the brain and the spinal cord, we also looked for CD4 T cell and CD4 ligand, which is expressed in the CD4 T cell. So to give an overview, like what happens when virus comes, how virus actually cause inflammation. So virus enters, it Then cellular sensors like toll-like receptors, RLR, rig-like receptors, they actually recognize the pathogen. They get activated and they actually activates the transcription factor IRF, NF-kappa B, and they induces the interference. Interferon induces several interferon regulating genes IASg54, and this IASg54 actually translated into IFIT2, which is interferon induced tetratychopeptide repeat proteins 2, and this protein has protective antiviral response, because when Dr. Ganesh Sen in Cleveland Clinic made the IFIT2 knockout mice, they showed in several viruses, in collaborating with several faculties at different institutes, that West Nile virus, mouse hepatitis virus, vesicular stomatitis virus, Sendai virus, whoever you can infect with the mice, these mice are very much susceptible. So that they showed that the IFIT2 has antiviral properties. But to understand how it provides the antiviral properties, we studied in IFIT2 knockout mice. We inoculated with only 2000 PFUs. Generally we give 20,000, but mice are very susceptible, so only we did 2000 PFUs, and we looked at gross neuropathology. As I said, my lab is mainly focused on neuropathology, viral titer, immunophenotyping, immunohistochemistry and flow cytometry, as well as cytokine and chemokine array. And combining this data, we showed that the virus in IFIT2 knockout mice produces severe clinical score, means clinical diseases. The mice cannot walk properly, they have hind limb paralysis, and we have an arbitrary scale to give the score. You can see in wild type mice it is not more than 1, but in IFIT2 knockout mice it is more than 2.5, almost close to 3. So as a cause, we tried to see why it is so. We saw that there are significant amount of viral titer is present both in the day 5 and day 7 post infection. And as we use the EGAP tagged virus, by EGAP you can see the virus is propagating or spreading throughout the brain parenchyma much more compared to wild type mice. So at that point we thought virus is replicating more, so we should get more inflammation. But surprisingly, we observed that while wild type mice, as I showed you, it causes meningitis, inflammation in the meninges, then perivascular cuffing, microglial nodule, IFIT2 knockout mice are much more quiet and silent. There is not much activation of microglia macrophages. So IFIT2 deficiency decreased viral induced cellular inflammation and microglia macrophage activation. So that is basically telling like why, due to lack of inflammation, actually mice were dying even at 2000 PFUs. So if it is in the acute phase, what is happening in the chronic phase? So we used only 500 PFUs because mice were not surviving after day 8 at 2000 PFUs, and we looked at it even at 500 PFUs. At that much low PFU, the virus in IFIT2 knockout mice developed severe clinical score, and there is a significant weight loss, and the viral RNA particle which is supposed to be resolved by day 5 to 7 post infection still persists. So virus is not cleared completely, and as virus is not cleared, they causes severe demyelination. Compared to wild type, very little demyelination observed, but in IFIT2 knockout mice we observed a large amount of demyelination. So if we show that IFIT2 deficiency mice showed severe weight loss, heightened clinical score, sustained viral persistence, and severe demyelination at day 30 post infection compared to wild type mice. So again we started asking why it is so. Then we observed that in the demyelinating plaque there are lots of activated monocyte microglia macrophages. At acute stage, surprisingly, we didn't see any microglial activation, but we showed that T cell couldn't migrate, and as T cell is not there, this monocyte microglia macrophages could not bring back its pro-inflammatory to anti-inflammatory condition, and still the phagocytic macrophages are present. Compared to wild type, you can see a large number of phagocytic microglia macrophages are sitting there. And when we use the electron micrographs, we showed that this microglia macrophages, they are sitting in the demyelinating plaque, and you can see here that microglial cell membrane is in intimate contact with the outer portion of the myelin sheath, and here you will appreciate that they are microglia macrophages are producing pseudopodia and stripping the myelin. So the myelin stripping, microglia macrophage mediated myelin stripping is one of the cause of viral induced demyelination in fit-to knockout mice. That's what we concluded. And then from these two studied, we came to a point both at the acute stage in the brain and chronic stage. This paper just got accepted in Journal of Virology, and that in 2020 PLOS pathogen paper, that IFIT2 provides antiviral function by promoting acute neuroinflammation and thereby aiding viral control and limiting severe demyelination. Whereas reduced infiltration of CD4 T cells at acute stage of infection leads to phagocytic phenotype of microglia at chronic stage, leading increased microglia mediated myelin stripping and thus demyelination. So from the mechanistic standpoint, we came to a point that microglia monocyte macrophages myelin mediated myelin stripping is major cause of the IFIT2 mediated viral clearance and viral myelin stripping. So then if I combine these two, we came to two dyads. One is the T cell, CD4 T cell, and one is microglia macrophages. CD4 T cells migrated from the periphery. Some of microglia were resident, but some of the macrophages came from the periphery too. So we wanted to dissect out what is happening at the molecular level. So CD4 T cell expresses CD40 ligand. CD40 ligand binds to CD40 in macrophages and gives the downstream signaling to secrete TNF alpha, IL-1, IL-6, and these chemokines for infiltration of immune cells helps the more T cell to come and T cell to provide help, to provide aid to clear the virus. So that's what we are looking for the diet. So first what we did, we used the CD4 knockout mice, and we showed that in CD4 knockout mice there is a grey matter inflammation, poliomyelitis, DRG inflammation, profuse demyelinating plaques and abnormal bulbar vacuolation. Whereas when we use the CD40 knockout mice, we observed that CD40 mice makes mice more susceptible and dampens microglia macrophage activation and causes severe chronic demyelination. So CD40 ligand deficiency result in severe chronic stage demyelination and axonopathy also. So by taking this diet CD40 ligand interaction, we can conclude that MHV induced demyelination and axonal loss are not primed by the adaptive immune system which happens in the EAE, CD4 T cell mediated damage, with its array of specialized T cell and made to order antibodies produced through complex gene arrangement. Rather, it is innate immune system upon which the adaptive immune system is built, and that's what when we concluded. This is not only two studies, these are also our publication which supports this study, and our lab also works in different aspects, like I just expressed some of this data in neuroimmune modulation, but we work in the structure biology of the structure function analysis of the spike protein, antiviral therapeutics, genomic control of pathogenic properties, host cell response and cell-cell communication. Also, like as I have a long-term experience to work with the mouse experimental model system, I was bestowed with a grant to develop the cancer model for cervical and ovarian cancer to test therapeutics by System Medicine Cluster Project through NIBMG. And I am thankful for these studies. My 13 PhD students who graduated, they significantly contributed in my studies, and now I have 15 graduate students who are pursuing PhD. They are also significantly contributing, and this work was not possible if Professor Ganeshan didn't give me the IFIT2 knockout mice. I am thankful to Cornelia Bergman, also Cleveland Clinic, for her insights. Maria Nagel, Ravi Mahalingam, Kenneth Schindler, Michael Koval, David Bloom and Ujjwal Niyogi. We have a group of people which we are working as a virtual network project in IUSSTF, and my collaborator from IISc, Dr. Demyelinat Pal, Arup Banerjee from RCB and Dr. Vidisha Sinha from IISER Kolkata. And at the end, I want to thank funding bodies. Without their help, the work was not possible. CSR, IUSSTF, IISER Kolkata, of course, SCRB, ASM, American Society for Microbiology, High Media, DBT, and National Multiple Sclerosis Society, USA. And thank you for your patience.
Thank you very much for finishing exactly in time. It's a brilliant presentation. So I'm sure there are some questions. Well, before anybody asks, so you said that the European population shows much greater susceptibility than, so are there a genetic factor?
Genetic.
And some of those, could you elaborate on?
Yeah, that is MHC, major histocompatibility complex, DRBQ motifs. They are very much susceptible for infection, and that's what people believe, that they have a genetic background, and then when the infection comes together, they exhibit the disease process.
So is age also a factor?
Age is a factor, because the disease starts at the age of 20 and it goes until like 40, 50, and after that people goes to wheelchair.
No, so the population where it is more sensitive and the one where it is not, is also the age a factor? I mean, the one where it is more sensitive.
Yeah, of course, the age is a factor too.
Thank you very much. Such a brilliant presentation.